welcome to Anthropology. Today's topic is abnormal hemoglobin, which include hemoglobin E and hemoglobin F. Firstly, we focused on hemoglobin E. It was first identified in 1954 in a patient, and this patient was inherited that gene from his father, and his father was of Guatemalan origin with Spanish and Hindu ancestry. In the same year, that means 1954, another case was identified in Thailand. Okay. Hemoglobin E variant is a polymorphic trait and considerably important for Asian anthropology because it is frequently seen among the people of Southeast Asia and associated with Mongoloid people. Okay. So these are some important short questions also. Next. J.B. Chatterjee in the year of 1950 first reported it in India. That means in 1950, the first case of hemoglobin E variant was identified by J.B. Chatterjee. And it was frequently seen among the Toto people. Toto is a tribe of Northeast. So Toto people, and the frequency is 23%. Genetically, in the case of hemoglobin E, it is the cause. Okay, it is the cause. The glutamic acid at the position of 26 is replaced by lysine in beta chain. And it is not genetically so severe, but used as a marker for population discrimination. So hemoglobin E variant is not so severe genetically, but useful as a marker for population discrimination. Okay. And highest frequency of hemoglobin E was reported from Assam also. Next. Here is a chart. Some tribes and frequency of hemoglobin E. Khashi 0.21%, Kachari 0.50%, Garo Rava tribe 0.49%, and Mikir 0.19%. Okay, very important. Please note down it. Next, hemoglobin S. In 1901, a Chicago physician first reported it. A presence of a peculiar elongated sickle-shaped cell in the blood of an anemic student of African ancestry. So first it was identified by a Chicago physician in the year of 1901. And the shape of the blood cell was elongated sickle shaped. Soon it was found that such sickle cells are quite common among Negro. So hemoglobin E common among Mongoloid, associated with Southeast Asian, and hemoglobin S variant frequent among Negroes. Moreover, it was noted that the type of anemia is hereditary. Okay, so it is a hereditary disease. Sickle cell anemia is a molecular disease and it's produced by presence of an abnormal hemoglobin gene, obviously called hemoglobin S. Okay, next. Patient with sickle cell anemia are homozygous for the gene. So this is very important. Homozygous. Patient with sickle cell anemia are homozygous for the gene, while in heterozygous condition is of milder order. So severe in homozygous form. Homozygotes for hemoglobin S show severe clinical symptoms like enlarged spleen, rheumatism, impaired mental function, also called mental retardation, okay, besides anemia. Among them, mortality rate is high. People who are heterozygous in genotype are not suffer from severe breathing problem for heterozygosity. Okay, and they also not affected by malaria. So people with heterozygosity are free from malaria infection. Okay, like people with normal hemoglobin. So if the condition is homozygous, then it is very severe or harmful or deadly. But If it is heterozygous condition, it is sometimes advantageous. Okay. Here is another chart, gene free percentage and some region. So central region tribe Santal, western region Vil, southern region Toda, Kurumba, and the gene percent is 5 to 10%, 10 to 15%. For central region Muriagond, Rajgond, western region Vil, and southern region Chenchu, and the north and northeastern zone no data is collected. Okay, 15 to 20% Gond for central region, western region for Vil, and Kurumba for southern region, 20 to 25% for Garacia of western region and Irula from southern region. Okay. So these are some information about hemoglobin E and hemoglobin S. Okay, that's for today. Thanks for watching. Please subscribe our channel Anthropology.